Hello everyone, we are ready with our new video on the disease tuberculosis. In this video we will cover the different stains causing TB, the pathogenicity, the different symptoms, the infection cycle, diagnosis and treatment of the disease. Now we all know that TB is a chronic infectious disease which is caused by a bacteria which mainly affects lungs, but it may involve other organs like brain, kidneys and spinal cord. Early diagnosis and proper treatment, they are essential to prevent the spread of the disease and drug resistance. Now, for the very first time, Robert Koch identified Mycobacterium tuberculosis as the causative agent of TB. And in addition to Mycobacterium tuberculosis, worldwide, Mycobacterium bovis and Mycobacterium africanum, these are the two strains which are found in addition to Mycobacterium tuberculosis. Now, as far as the pathogenicity is concerned, the infection results when the bacteria is phagocytosed, it is engulfed by the macrophages, which are the immune cells in the lungs. Now, the Mycobacterium tuberculosis, when it is phagocytosed, when it is engulfed by the macrophages, they reside inside them or they survive in them by normal antimicrobial process. So the infected macrophages, they often die attempting to destroy the bacteria, and within 4 to 12 weeks the viable bacteria, they are released into the respiratory spaces and thereby developing the disease. It involves various symptoms like fever, fatigue, weight loss. Now, if I talk about the infection cycle, the Mycobacterium tuberculosis is inhaled and through the respiratory tract, it enters the lungs, as you can see here in the diagram. And when it enters the lungs, it is engulfed by macrophages, engulfed or phagocytosed by the macrophages, and it may result in latent tuberculosis or it may result in which later develops into active tuberculosis. And latent tuberculosis is somehow dormant. So if I talk of latent TB, the immune system controls the infection, means the macrophages, they will not spread it to the nearby cell, and the intact tubercles are formed and bacteria are alive inside the macrophages but dormant, they are not spreading. So this condition of latent tuberculosis, it is asymptomatic. While if I talk about the active tuberculosis, here the immune system fails or the individual is immunocompromised or it is weak, and the dormant bacilli which are residing inside the macrophages, they reactivate and they start to multiply. The granuloma, they will break down and the tissue damage will occur. There will be chronic cough, fever, means there will be disease progression and there will be potential reactivation. Here I have used the term granuloma, which are the organized collection of the immune cells that contain the bacteria, means your macrophages are your immune cells, your T cells are your immune cells. So all these cells where your bacteria is also present along with them, that we call as granuloma. Now, once the active TB have been developed by the multiplication of Mycobacterium tuberculosis in the cells, these are the symptoms like fever, fatigue, weight loss, cough, and these conditions finally results in the bloody sputum. Now, what gives mycobacteria, what are the different features of mycobacteria which help it to cause the disease, which plays roles in its pathogenicity? Here in the diagram you can see that the cell wall is the main thing which helped it to do so. There is a thick lipid rich waxy cell envelope which enables Mycobacterium to resist the host defenses and persist inside the macrophages. They can avoid the lysis inside the macrophages. Otherwise, macrophages are the cell which can kill the entering cell, which kill the engulfed bacteria. But here they are not able to do so. In addition to this, the cell envelope of Mycobacterium, they contain unique lipids. As you can see in the diagram, there are lipids, there are glycolipids. Uniquely, they have mycolic acid, they have lipoarabinomannan, which inhibit macrophage activation and interfere with phagosome maturation. There is trehalose dimycolate. These glycolipids and the lipids, they are directly toxic to the eukaryotic cells, and they creates a hydrophobic barrier around the bacterium. Means they do not let the enzymes, they do not let the antimicrobial substances to work on this bacteria, and this facilitates a kind of impermeability and resistance to the antimicrobial agents. There it could be an acidic or the alkaline compound, osmotic lysis, lysozyme. All these host defense mechanisms, they are not able to perform on the Mycobacterium. Now these are the different advantages. Here you can find them listed, that these are the different advantages of membrane lipids and glycolipids. They are directly toxic to eukaryotic cells. These glycolipids, as we discussed them so far, they create a hydrophobic barrier and facilitates impermeability and resistance to antimicrobial agents. So they can easily associate with the mannose residues which are present on the eukaryotic cell surface. So we all know that eukaryotic macrophages have mannose residues on them, and in the form of mannose receptor, macrophage mannose receptor, or we call them as MMR. And binding of these mannose rich ligands to the receptor, it facilitates the endocytosis and the phagocytosis of Mycobacterium into the macrophages. Now see how the tuberculosis progression, it takes place inside the human lungs. The Mycobacterium tuberculosis survives in macrophages. It establishes over there, and it resists the oxidative damage. It inhibits the phagosome lysosome fusion, which is the main step wherein the bacteria can be killed, but it inhibits that phagosome lysosome fusion, which results in granuloma formulation. As I told you, granuloma, an organized collection of immune cells that contains the bacteria, and thereby forming the necrotic core. Necrotic core is formed due to the prolonged immune response and lack of oxygen. It contains the dead host cells and the bacteria, and giving it a cheese-like appearance. And once the necrotic core has been formed, there has been the formation of tubercles in the lung. Now here you can see that the tubercle lesion, which has been formed in the lungs, if it liquefies, it forms the air-filled tuberculous cavity, and from where the bacteria can be disseminated, it can spread. That is called miliary tuberculosis, and the name is due to the tubercle size, which is of millet seed size in the infected tissue, wherein the tubercle lesion, it has a cheese-like consistency. If it calcifies, then Ghon complexes are formed, which are observed prominently in X-rays. If I talk about the diagnosis, we have three main diagnosis methods. First are the clinical features. As we all know, clinical symptoms we can say. So persistent cough is there, hemophysis is there, fever is there, weight loss is there. Hemolysis is the bloody sputum, presence of bloody sputum. Laboratory tests include the sputum examination by Ziehl-Neelsen staining, which is detected by which detects the acid-fast bacilli of Mycobacterium tuberculosis. And immunological test is also included in laboratory test, which includes the Mount-X test, and in Mantoux test, purified protein derivative, in short we call it as PPD, of Mycobacterium tuberculosis is injected into patient and the patient, it shows the various symptoms, means there is a hypersensitivity which occurs within 48 hours. There is hardening, there is reddening of the area around the injection site. And finally, we have the radiological findings where the chest x-ray, it shows the nodular lesions of tubercles, and it is required to be verified with the other diagnostic test. Now, if I talk about the treatment, there have been antimicrobial therapy. So TB is curable with this antimicrobial therapy, which we call as HREZ Rezyme, which involves the various antimicrobial medicines like isoniazid, rifampicin, ethambutol, and pyrazinamide. They are given together as HREZ Rezyme. Second, we have the DOTS therapy, which is also attained by WHO, which is directly observed treatment. It is a short course treatment where patients, they take these antimicrobial drugs under the supervision, and there is the less chances of developing resistance. So in DOTS, which is a WHO strategy, it ensures that TB patient take their full 6 months medication course under the supervision, and there will it is ensured that there is a proper drug supply, monitoring, and direct observation by health workers. So these are the two main treatment strategies. So with this, we are done with the topic. Thank you for listening. Please like, share, and subscribe. Thank you so much.